Hello, good afternoon. We're team number 2 and it is made up by Sara, Brayan, Josué, David and Pilar. Today, we are here to explain you a little bit about the amazing DNA. Hope you enjoy it, but learn. The clips you're observing right now show the process we've been through for building our final project. Keep watching so you can see it. It was made on Minecraft, a very famous video game where you can work in team and you can build such incredible things just like this DNA.
Our purpose is to explain and show the documentary investigation of the social and scientific implication of DNA model proposed by Watson and Crick, and a 3D model of the DNA molecule. We investigate about the structure and function of this molecule as a carrier of genetic information, as well that the observable nucleic acid, also known as DNA, is made up of molecules. Receive the term of polynucleotide because it contains several nucleotides that are made of three parts: a phosphate group, a sugar group, and one four types of nitrogen bases. The information of this molecule is stored as a code made up of chemical bases.
Okay, now I'm going to talk about the structure and chemical composition. We know that the DNA is made of chemical building blocks called nucleotides. These building blocks are made of three parts: a phosphate group, a sugar group, and one of the four types of nitrogen bases. To form a strand of DNA, nucleotides are linked into chains, with the phosphate and sugar groups alternating. The complete DNA instruction book or genome for a human contains about 3 billion bases and about 20,000 genes and 23 pairs of chromosomes. The four types of nitrogen bases are set up in nucleotides and its characters are A, T, G, and C. The order or sequence of these bases determines what biological instructions are contained in a strand of DNA. For example, the sequence ATCTGT might instruct for black hair, while ACTGCT might instruct for blonde. The molecular structure of the four nucleotides is very similar, because it is composed of the three parts: a base containing nitrogen, a phosphate group, and a pentose of 5-carbon sugar. Only the base changes according to the type of nucleotide. The nucleotides adenine and guanine have 2 carbon rings and are called purines. Then the thymine and cytosine nucleotides have only 1 carbon ring and are called pyramides. The strands of DNA are formed by the covalent bond between the phosphate group and the hydroxyl group of a sugar, and these steps are formed by the bases that are coupled together. It means it has a structure in which two complementary chains join. Their nucleotide function as a small block that, when joining together, form two long chains of DNA. Important rule between notes as grammatical: the four characters can be written in any order, as long as adenine is always paired with thymine, as cytosine is always paired with guanine. The adenine and guanine are large molecules cause their carbon rings are fused. In contrast, thymine and cytosines are small, as they are formed by a single ring. The cells in your body constantly divide, regenerate, and die, but for this process to occur, the DNA within the cell must be able to replicate itself. During the cell division, the true strand of DNA is split, and the two single strands can be used as template to construct a new version of complementary strand. As A always pairs with T and G always pairs with C, it is possible to work off the sequence of bases on the one strand using opposite strand, and it is that allows the DNA to replicate itself. The process is carried out by a family of enzymes called DNA polymerases. DNA is composed of two side-by-side chains of nucleotides twisted into a shape of double helix. The two nucleotide strands are held together by weak association between the bases of each strand, forming a structure like a spiral staircase. The backbone of each strand is repeating phosphate, deoxyribose, and sugar polymer. The sugar phosphate bonds in these bicones are called phosphodiesterase bonds. Finally, the attachment of the phosphodiesterase bonds to the sugar groups is important in describing the way in which a nucleotide change is organized.
DNA functions. As it was mentioned before, DNA is the structure that contains the instructions needed for an organism to develop, survive, and reproduce. For carrying out these functions, its sequence must be converted into message which can be used to produce proteins, which are the complex molecules that do the most of the work in our bodies. However, there are other tasks that the DNA develops, such as the following ones. Replication. DNA exists in a double helix, in which each base along one strand binds two complementary bases on the other strand. When a cell divides, the chromosomes containing the DNA strands must replicate so that both daughter cells receive the full set of genetic material. During replication, the DNA double helix unwinds, allowing each strand to act as a template for a newly synthesized complementary strand that forms a double helix.
Replication, as it was mentioned, helps DNA to make up proteins we need for living. However, there are also some other functions that this structure develops, together with other ones just like the RNA or enzymes like helicase, polymerase, and even unions of chromosomes. Deoxyribonucleic acid also encodes information, mutates, recombinates, and expresses genes. From all the functions, we consider the making of proteins as the most important, and it takes place in two steps: transcription and translation.
After all the information we have explained and analyzed, we can conclude that DNA is just awesome, and although it's a small molecule, it has a lot of power. As we must know, we can locate all of the terms that we saw and mentioned in a big scientific field, genetics. It is the study of how traits such as a hair color, eye color, and risk of disease and inherited from parents from their children. Also, it is worth to mention that genetics influence in how these inherited traits can be different from person to person. And pointing this, we can see the huge importance of it. DNA transcripts, encodes, mutates, but specifically it replicates, ensuring that each daughter cell from at the end of cell division receives equal amount of DNA. This structure is important in terms of heredity. It packs and in all genetic information and passes it on to the next generation. The basis for this lies in the fact that DNA makes genes and genes make chromosomes, and we humans have 23 pairs of chromosomes. 22 of these pairs, called autosomes, look the same in both males and females, but the 23rd pair is called the sex chromosome. Females have copies of the X chromosomes or double X, while males have one X and one G chromosome. Now, imagine that DNA isn't copied. A lot of problems will evolve. For example, the daughter cells won't receive all necessary genes, and there may be absence of important proteins which are encoded by DNA. Then, once more, genetics is what makes life possible. It's the thing that makes us unique. By including genes which indicate our body cells how to grow and function. And these ones are like bricks for a building to body foundation.